Hello, and welcome to this brief tutorial on the COVID-19 crowd-generated gene and drug set library created by the Mayan Lab at the Icahn School of Medicine at Mount Sinai. This crowdsourcing website allows scientists and researchers to submit COVID-19 related gene sets and datasets, along with metadata for their work. Datasets are manually reviewed before being published and hosted in the database. Researchers can explore the hundreds of gene sets and drug sets to aid their own research in a global effort to combat COVID-19. Let's take a closer look. At the top of the page are the current statistics of datasets and gene sets in the database. By clicking either the Unique Drugs or Unique Genes links, you can see a total count of mentions of each drug or gene within the submitted sets. You can also click the Top 20 link to generate a bar graph of the top 20 genes or drugs. Let's return to the home page. You can choose to explore either drug sets or gene sets. In addition, the drug sets have 4 categories to narrow your search: drugs from biological experiments, computationally predicted drug sets, drugs mentioned in Twitter in a COVID-19 context, and all categories combined. You also have the ability to search for a drug or gene using the search bar here. Regardless of whether you choose to explore the drug sets or gene sets, both will provide you descriptions of the dataset by clicking on the set name, including the source of the dataset, date of publication, and possible other metadata. Clicking on the number of elements shows a preview of the members in the set, and an option to view the drug set in more details. On this page, you will see the description and other details of the dataset, plus the full set of elements. Clicking on any of the elements will bring you to a new page displaying all datasets the element has been listed in, plus a link to either Drug Bank for drugs or Harmonizome for genes. Finally, you can use the Venn diagram feature to compare datasets and look for overlap of either drug or genes. Simply select the datasets you want to compare, then click the Draw a Venn diagram button. Hover over the diagram to list the elements. You may also submit your own dataset. At the bottom of the home page is the submission form. First, select whether you're submitting a drug set or a gene set. Enter the relevant information about your dataset, including the dataset name, URL to the source, and background information on your research. Paste your gene or drug set in this text field. Please remember to keep each element on its own line. If submitting a drug set, please indicate whether it belongs in the computational, experimental, or other category. You may also add metadata information. Click the Add Metadata button and 2 text boxes will appear: parameter and value. You may add any parameter and matching value as you wish, such as organism and tissue type. To add another metadata field, just click on the Add Metadata button again until you have the desired number of fields. Finally, please include your contact information: name, affiliation, and email. Your contact information is hidden by default. If you would like it visible, please check the Share Contact Information box. When complete, click the Submit button to submit your dataset. If you have any issues or discover a bug, please report the issue using the Submit an Issue button at the bottom of each page. You may also share the homepage or any individual drug or gene sets to social media. A collection of social media buttons are available at the bottom of every page. Thank you so much for following along with this tutorial. We hope you found the COVID-19 crowd-generated gene and drug set library database helpful in your research. Thank you again, and we wish you the best of luck in your future studies.